So see if anybody's awake. In any case, the location is what you want to measure. So our individual molecules, in our microscope now, the bar is 250 nanometers. It looks like a little mountain if I plot the signal in the z direction, and I want to find the position of that molecule. So what we do is to fit the shape. We've measured the shape in the microscope, and we use a function, a Gaussian function, and others to determine the position of the molecule. Then so for example, in 2 dimensions, I'm showing the image of the molecule. With some fitting of data, we find out where it is. So that's the first key idea. And this works to a precision that is the Abbe limit divided by the square root of the number of photons detected. If I detect 10 to the 4th photons, then I can localize to 100 times below the diffraction limit. Instead of 200 nanometers, in principle, it would be 2 nanometers in terms of the position. But this only works for one molecule when it's separated from all the others. And this is not resolution, it's only localization. So to get resolution, you have to do one more thing, and I like to call that active control of how many molecules are on at any given time. So the idea is we have a structure we want to see, and you put fluorescent labels all over it. If you allow them to all emit at the same time, then you get this very blurry, fuzzy picture. And so that's the idea. You don't let them all emit at the same time. You actively make sure that only a few are on. Using an on-off process, using either photochemistry or blinking, like I was just describing a moment ago. I start out dark and then only allow a few to emit. Then I can localize those with the upper trick and then photo bleach them, and then you localize another group randomly selected, photo bleach them, and you keep writing down these positions of all these molecules. In the end, you take all the positions and show them at the same time, and you see structure beyond the diffraction limit. So that's how it works. And this has been called many names with many acronyms. I won't go through all of these acronyms, but a mechanism-independent acronym that we like a little bit is called Single Molecule Active Control Microscopy, or SMACM. All right, so that's how it works, but let's try one more time to make sure everybody understands how this works. If you will, maybe you can explain this to your children or your family. Think about fireflies for a minute. Fireflies. You've all heard of fireflies, right? People have heard of fireflies. Yes? Yes? Got to show. So now imagine you have a tree. You have a big tree, and the tree, but it's nighttime, and you want to see the branches of the tree. So it's dark, though. You can't see them. So the idea is to place fireflies all along the branches of the tree. Okay? Place fireflies. And then just take your camera and take a movie of what's happening, because the fireflies will blink on and off, randomly at different positions. Every time you see one of them on, you measure where is it, and write that down. Measure where is it, write that down, and then show them all the positions at the same time, and you'll see the branches of the tree. And that's how this works. And it uses those single molecules. So I'll show this to you a little bit differently here with molecules, the same idea. On the left is the diffraction-limited image, one frame of the movie. This is now a mammalian cell, and we've labeled microtubules, and you see these dots, which are the single molecules from one frame of a blinking movie. And on the right, you see little tiny dots, which are the locations of all those molecules. Next frame of the movie, remember it's blinking. Next frame of the movie, blinking. And here, if you just run the movie, try again. There. You see that from all of that blinking, you get the information about the positions of the molecules and this image with very high resolution. So that's the idea. Now, as chemists, you want to think more about this process that lets us control the molecules, having them be on and off. And so I don't look at the long list of mechanisms that have been demonstrated. Just look at these energy levels for a moment. Remember, we want to create a fluorescent object that you can pump and emit in the middle. That's what you want to create, but in low concentration. So one way to do that is to start with a dark precursor molecule, a molecule that's not emissive, but then maybe a little control light will convert a precursor into an emissive molecule. And if you do that at low enough optical intensity, you'll create just a few of those molecules at any given time. That's one way to do it. Another way to do it is this blinking business, and blinking comes from an emissive process that's coupled to dark states on the far right. And so those dark states on the far right act as reservoirs to hold the molecules in the dark so they do not emit. And if you put most of them into these dark states, then every once in a while one will return and you'll see it emitting again. So this is what blinking is coming from a variety of processes, for example, photoreduction of the molecules with certain additives. And there's other mechanisms. Even to give you a little more flavor for the diversity in this area, but the need for more ideas, on the top I'm showing you a precursor molecule that's converted to a fluorophore. That's because if you see the aromatic azide on the bottom left, that's not a very good donor in this donor acceptor molecule. So a little irradiation in the blue will convert that azide. It will be basically blow off dinitrogen. There will be reactive nitrene left behind, and you get an amine. Not exactly the structure shown, but you can produce an amine from an azide this way, and now you turn on fluorescence. So that's been demonstrated. Another scheme is to label a molecule that you would call a ligand that binds somewhere that's important, like this molecule, saxitoxin, which binds to voltage-gated sodium channels on the surface of cells. Now you allow the molecules that are moving around in solution, you don't see them very well because they're fuzzy, because they're motion. But then when they bind and sit on a channel, they sit at one place and you can localize the molecule that way. It's a technique called paint. Another example, here's a rhodamine spirolactam that can be opened by irradiation, which converts a dark molecule into an emissive molecule, and so on. So, but I want to say that there's a continuing need from our synthetic friends to come up with more molecules that we can control in terms of whether they're emissive or not. Okay, so now the next signpost. This super-resolution fluorescence microscopy is fascinating because it gives us a new window into the nanoscale. Because we're looking on the scale of these single molecules that are about a nanometer in size, and we can extract information based on how many photons we can collect from each molecule. The idea required fundamental scientific advances, uncovering this issue of can you detect a single molecule at all? And then what can you do with it? But also it required these surprises, the blinking and switching that I talked about, which lets us control the molecules. Examples now. Let's just enjoy a few cases. These are bacterial cells. 3 different proteins inside these bacteria have been labeled by fusing enhanced yellow fluorescent protein, EYFP, to the proteins of interest. Here's the diffraction-limited images. Okay? The diffraction-limited images don't tell you very much, but the super-resolution images are spectacular. The difference is just wonderful. With an object as small as a bacterium, it's already close to the diffraction limit in size. So you can see a lot when you just go below the diffraction limit by a factor of 5. And so that's one example. Another class of examples, and there's just too many for me to review because there's hundreds now. This is a case of mammalian cells in part of these pictures. On the upper left is a spectacular demonstration of something that was discovered by this super-resolution microscopy, which is the banding, the periodic banding of the spectrin molecules in an axon from the Zhuang lag. On the lower left are the very small aggregates from Huntington's disease. The amyloid diseases all have very small fibrils and aggregates that form inside cells. And many of the sizes are below the diffraction limit. This is actually observed with STED microscopy because we have also built a STED microscope. The upper right is bacteria again, but we're looking at something called the S layer that forms on bacteria and doing pulse chase, where the old S layer is in green and the new is in red. And with super resolution, you can see a great deal of detail about how this layer is formed. And the lower right, glycocalyx. The glycocalyx, something that many people haven't heard about. But all of our cells in our body almost are covered with sugars, covered with different glycans in different patterns. And these glycans are important for recognition between cells and interacting. They're elaborated in cancer and so on. And this is a super-resolution microscopic image of the surface glycans that have been detected by using methods from Carolyn Bertossi to attach fluorophores on specific glycans. You can see that they're hollow tubules. They're extending out from the membrane are these hollow tubules that form. And you can observe the shapes of these structures.